Welcome to Biology Made Easy. Today we are discussing experiments on respiration. In our last lesson on respiration overview, we discussed these terms extensively: aerobic respiration, how glucose is oxidized oxygen to produce carbon dioxide, water, a lot of energy, 38 ATPs and heat, and how anaerobic respiration, known as alcoholic fermentation in yeast and germinating seeds of plants, uses glucose without oxygen to produce ethanol, carbon dioxide, energy, heat plus only 2 ATPs. And anaerobic respiration, known as lactic fermentation, goes on in some bacteria and skeletal muscles of mammals, where glucose is broken down without oxygen to lactic acid, 2 ATPs, and heat. Now, in today's work, we want to look at the experiments that prove that respiration has taken place. And the factors that can be tested are oxygen absorbed, carbon dioxide evolved, and heat evolved. Now, one will ask, why don't we test and prove that glucose has been used up or water is produced? It's difficult to find out how much glucose is used up in an organism, and it's also not wise to find out water evolved during respiration, because even non-living things, when they are wet, has water. So, experimenting respiration, we want to look at major chemicals, tools, and their function, which we would use to prove oxygen absorbed, carbon dioxide evolved, heat evolved. We have chemicals: caustic soda, sodium hydroxide, soda lime, potassium hydroxide. These absorbs carbon dioxide. Lime water, calcium hydroxide, is an indicator, turns milky when carbon dioxide is present. Pyrugallic acid absorbs oxygen. Hydrogen carbonate indicator, it's red, it turns purple when there's very little carbon dioxide around, and it turns orange or yellow when there's plenty of carbon dioxide around. Then we have dichlorphenol, indophenol, DCPI, or methylene blue. This is an indicator that turns from blue to colorless. It's used to show the rate of respiration in some substrates. And then you have a tool called manometer. It's a U-tube and it contains liquid. It is used to indicate pressure differences in a closed system. So if pressure changes in a system, the manometer shows by showing some differences. The manometer contains fluid, and the fluid can be a dyed water, oil, or mercury. The lighter the fluid, the greater the changes in the manometer with little differences. It shows a great change. Then we have a scale that is attached to the manometer that can measure the differences that occur. And we have a respirometer. It measures the rate of respiration by measuring the rate of oxygen uptake, and the respirator is a setup that has manometer attached to it usually. Then we have other chemicals like formaldehyde or formalin. It's used in washing seeds to prevent bacterial and fungal growth. We have oil or paraffin that will cut off water. We have an instrument called the calorimeter that measures energy levels in respiratory substrates. Respiratory substrates, you know, is carbohydrates, fats, and protein. Those are the substances that are respired. Now, let's look at the experiments. The first one, we'll look at anaerobic respiration, and we'll look at energy or heat produced during respiration. This is a setup. This is a vacuum flask. Germinating seeds here in the vacuum flask. The germinating seeds are washed in formaldehyde or formalin to prevent bacterial or fungal growth. The cotton wool is sealing up the entrance to the vacuum flask to make it airtight and also to hold the thermometer. You see the vacuum flask fits inside, all right? Now this setup, if it's left for 2 days, will produce enough heat, all right? That can be measured for control experiments, germinating seeds that have been boiled so that the seeds are dead. But then the dead seeds are also washed in formalin. So we also have this setup to prove carbon dioxide is produced during anaerobic respiration. Here, this is the setup. This clip or three-way tap is open, but just before the experiment starts, this tap should be closed. Now, this is yeast and glucose solution. Oil is poured on the solution. This oil cuts off air from the glucose and yeast solution. The yeast and glucose solution is prepared in such a way that there should be no air to start with. So water is boiled to get out the air, and the boiled water is used to prepare 5% solution of glucose and 10% dried yeast. That is what is here. Now, if the mixture is left for about 30 minutes with gentle warming, carbon dioxide gas will bubble out into this tube, and the gas bubbling into lime water turns the lime water milky with the carbon dioxide. Remember that the screw clip tap is closed at the beginning of the experiment. So the carbon dioxide that bubbled out cannot go out. It goes into this delivery tube, into this tube. The control experiment is just to use killed yeast, and the setup is the same. So we want to look at aerobic respiration. The first thing is oxygen is taken up during respiration. This is the setup. This setup is called a respirometer. It's going to measure the rate of oxygen uptake in this system. Now we have this as a test experiment tube. You have soda lime here, this gauze, these are wood lice in here, and this tube is connected to the manometer, that's the manometer, and connected to this system also. This is the control experiment. It also has soda lime here to absorb carbon dioxide, the zinc. You have these glass beads instead of the wood lice. The test tubes are put in a water bath, controlled water bath, so that the temperature can remain the same, because gas expands rapidly with temperature changes. So any small change in temperatures will show in the expansion of gas in these tubes, and the expansion of gas in these tubes will be shown in the manometer level. So the two systems, they put in the water bath the same temperatures. Now the experiment works in such a way that when the wood lice absorb oxygen, carbon dioxide is produced, and the carbon dioxide is absorbed by the soda lime. So the oxygen that is absorbed causes a reduction in pressure of this system here, and it shows in the manometer. And so we can read the differences in the height of the liquid in the manometer, and the differences in the height corresponds to the amount of oxygen that was taken up from the system, from the respiration. That is how this respirometer is used to show up that oxygen has been taken up, because the level of the manometer fluid, it will be higher here than here. We want to also note that at the beginning of the experiment, the screw clip is closed to have a closed system. This setup can be put together in different ways, so you should not get confused. You can have this one and the manometer. So the two are separated with the organisms and things inside it, and there's a scale here, a scale. So this is the manometer, and that is the scale. That is the scale. And these two setups here in the water bath. This is the test experiment. This is the control experiment. So the manometer, respirometer can be arranged in various ways. We can also have a simple respirometer. You have a glass container, you have the soda lime here, you have cotton wool, you have a gauze, you have your organisms, and you have a capillary tube here, and it has a scale, and there's a colored water in the capillary tube. This organism, blowfly larvae, absorbs oxygen in the system, produces carbon dioxide. The carbon dioxide is absorbed by the soda lime. It absorbs the oxygen for respiration, and so the pressure in this container reduces, and so atmospheric pressure presses here, and so the drop of water will move from A to B as the organism absorbs oxygen for respiration. This is a simple respirometer. We'll look at how we can use it to calculate the rate of respiration soon. Then we can also show oxygen is needed for germination or respiration in a simple experiment. You have these glass tubes. You have caustic soda here to absorb carbon dioxide. You have a zinc, you have wet cotton wool in here, you have your soak seeds here. This is the control experiment. In the test experiment, you remove both carbon dioxide and oxygen. The oxygen is absorbed by the pyrogallic acid. The carbon dioxide is absorbed by the sodium hydroxide, caustic soda. So this is the test experiment. These are soaked seeds, and soaked seeds germinate. So in this experiment, this is the control experiment, the seeds remove oxygen in the system, will germinate. Leave the system, it's a big system that will have air in. The experiment is left for 3 days, the seeds will germinate. In the test experiment where you have oxygen removed by pyrogallic acid, carbon dioxide also absorbed, the seeds will not germinate. We now want to show carbon dioxide is produced during respiration, and this is the setup. In this setup, air is pumped continuously in here and goes through all these tubes, containers, and there's a suction pump here that draws the air. That's how come air can pass through and pass through all this because of the suction pump. So the air comes into this container. There's soda lime that removes the carbon dioxide. The air comes into this. There's lime water to test whether the air here has carbon dioxide. If it's not milky, it doesn't have carbon dioxide. The air comes in here. In this bell jar, there will be an animal. If we are testing for carbon dioxide is produced during respiration in an animal, like a small animal, then the air comes in here. Because the organism respires, the organism produces carbon dioxide, and the carbon dioxide comes here and turns the water milky. That shows that carbon dioxide is produced during respiration. If you are using a potted plant instead of an animal in this bell jar, this potted plant, you have the whole bell jar covered with a black cloth to cut off photosynthesis, and then you have the potted plant, you have the pot also covered with a polythene bag to cut off carbon dioxide being released by soil organisms. In our next lesson, we will look at how these setups are used to measure the rate of respiration in various conditions, and then also how the respiration quotient is calculated using these instruments. So we'll meet in our next lesson. Please give us your likes and also subscribe. Thank you very much.